Hello friends, warm welcome to you all to the channel World of Zoology. In our previous lecture, we have finished the transgenesis method and now we'll start with the next, that is the methods of transgenesis, four methods, and now we'll start with the animal biotechnology, the cloning experiment for the preparation of the Dolly, the world's first clone mammal. Here the experiment is shown where the nucleus is removed from the somatic cell, that is a body cell of the fin dorsate ewe, which is shown here, and for that the cell is removed from the mammary gland epithelial cells. These cells are then cultured on a culture medium, and for that the medium control the cell cycle of the cell division, and G0 stage which was induced by inhibiting the growth of the cell. Then G0 cell was then fused with the enucleated ovule, which is obtained from the another adult Scottish black-faced sheep, and then this was aided by the short duration electric pulse, so that these two could get combine and form a structure as like that of the fertilized egg. That means when the zygote is formed, the activation of the egg takes place so as to form the embryo. Just like that, here electric pulse functions and it activates the ovum which is enucleated, that is artificially nucleus is removed from the egg, and the egg and this somatic cell which is derived from the epithelial cells of the mammary gland combine to form a zygote-like structure due to the electric pulses, and then it develops into embryo. That embryo is later on implanted into the uterus of the surrogate mother, and where the full-term development takes place into the body of the surrogate mother, and the embryo which is implanted into the surrogate mother, that mother gave birth to the Dolly. The entire story is here picturized in this slide. Now we'll start the actual process of the formation of the Dolly one by one. Dr. Ian Wilmot and his team of the researchers at the Roslin Institute, Edinburgh, Scotland in 1995 created a clone sheep called Dolly by a transfer of the nucleus from mammary gland, that is udar cell of the adult sheep. They took mammary epithelium of the udar, which is fully differentiated tissue, from 6 years old sheep fin dorsate ewe. It was placed in a culture medium and further grown. The culture medium controlled the cell cycle of the cell division, whereby the G0 state was induced by inhibiting the cell growth. The G0 cell was then fused with enucleated ovum, which is derived from the another adult Scottish black-faced sheep. This was aided by short duration electric pulses, which simultaneously induce cell fusion and oocyte activation. The fused cell was then kept in a culture medium where it underwent cleavage similar to that of normal zygote. Such an embryo was then transplanted into the uterus of the third sheep that acts as a surrogate mother or foster mother for its full-term development. Here remember that surrogate mother or the foster mother means the female which is artificially induced for pregnancy. Thus, Dolly was born and her birth was announced on 22 February 1997. This clone sheep had its genetic composition exactly like that of the parent from whom the nucleus was derived for the transplantation, and the nucleus was derived from the fin dorsate ewe. It has similarity with the sheep of whose cell it was, and so also with the it has no similarity with the sheep from which the egg is derived, and also with the surrogate mother. Thus, this method of reproduction could be genetically compared with the parthenogenesis, which is the asexual method of reproduction. As this embryo develops from an unfertilized egg and leads to the production of identical progeny, the success rate of this process is low. Here, remember that the development of the embryo takes place without actual process of fertilization, and therefore cloning process shows similarity with that of the parthenogenesis, which is asexual method of reproduction, and possessing the identical characters as like that of the parent from which nucleus is derived. But the success rate of the process is very low, and as it can be ascertained from the experimentation of the cloning of the Dolly, where out of 277 nuclei that were transferred only one at a time to each enucleated egg, only 29 eggs grew into embryo. Out of 277 nuclei transferred to the enucleated eggs, only 29 eggs grew into embryos, and from that 29, only 13 embryos could be successfully transplanted into the surrogate mother, and from that, out of these 13 embryos which are transplanted into surrogate mother, only one ewe was successful in giving birth to an offspring, and that was later named as Dolly. Here the story of the Dolly is shown in this picture again, which is summarized. The egg cell which is derived from the Scottish black-faced ewe, and this egg, after removal from the donor, is enucleated, and this enucleated egg is then fused or allowed to fuse with that of the mammary gland cell having nucleus, and then they are fused, that is electrically zapped, so that they could get combined or fused. Here remember that not only the fusion of the somatic cell from the Scottish black-faced ewe with that of the ovum which is enucleated and derived from the fin dorsate ewe takes place, but here the activation of the egg also takes place, as like that of the natural process of reproduction, where fusion of the egg and sperm results in the formation of the activated ovum that starts dividing immediately. Just like that, here the fusion of the enucleated egg cell with that of the somatic cell of the another animal, due to the when they electrically zap or due to electric pulses, it form a structure like that of the zygote, though it is not true zygote, and the combined cell now has a single nucleus from the nuclear donor, and it start dividing, and therefore the blastocyst is formed from that structure, and that blastocyst or the developing embryo is then placed into the uterus of the surrogate female, which is shown here as a surrogate ewe, for the further development, and the blastocyst later on develop into a fetus in about 5 months, and the lamb is born, which is named as the Dolly, which is the world's first clone mammal, is shown here along with the Dr. Ian Wilmot. Thank you for watching this video. If you like the video, do like, Share this video for the benefit of the maximum students and subscribe the channel World of Zoology for the further intimation of my next video on biotechnology. Thank you once